Hello friends, today we are going to learning reproduction in bacteria. In a plant and animal, the phase of growth first complete, then the plant and animals goes for reproductive phase. But in the bacteria, the growth phase and reproduction phase are taking place simultaneously. Bacteria reproduces by vegetative method, asexual method, and by a sexual method also. So reproduction methods in the bacteria, as I told you earlier, in a vegetative reproduction, the binary fission, budding, and fragmentation are the mode of reproduction. While in asexual reproduction, the formation of endospore and conidiospore are the mode of reproduction. While in a sexual reproduction, the bacteria undergoing the methods like conjugation, transformation, and transduction like methods. So, first of all, we will learn the vegetative reproduction. So, vegetative reproduction is very common in the bacteria and it is taking place by the binary fusion, budding, and fragmentation. So first of all, binary fission. So look here, this is a very common method. Look here, the single bacteria transversely divide into two bacteria by formation of a septum and the division of a nucleic acid takes place. A single bacteria divide into two by replication and duplication of nucleic acid. Then it forms the septum and transversely divide into two daughter bacterial cell. This is called as binary fission. यामध्ये काय होतंय तर एक बॅक्टेरिया जी आहे त्या बॅक्टेरियामध्ये जो डी एन ए आहे न्यूक्लिओइड आहे तो रेप्लिकेट होतो डबल होतो आणि मग तो स्प्लिट होतो डिवाइड होतो न्यूक्लिओइड म्हणजे न्यूक्लियस आणि न्यूक्लिओइड डिवाइड झाल्यानंतर त्या सेलमध्ये एक सेप्टम तयार होतं एक भिंत तयार होते की ज्यामुळं एका सेलपासून दोन बॅक्टेरियल सेल तयार होत आहेत. द नेक्स्ट मेथड इज बडिंग. दिस इज अ व्हेरी रेअर मेथड of reproduction. In this method, the bird get formed to the lateral branches. So bird-like structure get formed, and in that bird, the genetic material get transferred. Later on, this bird get detached from the parental bacteria and works as a bacteria. So this is called as a budding method, and the bacteria are called as budding bacteria. So very few examples are there of a budding bacteria. These are the Pasteurella, Blastobacter, Rhodosudomonas acidophili, like a bacteria showing the budding method of reproduction. And the last asexual method is fragmentation. In this, the filament of the bacteria breaks into the several fragments, and each fragment now works as a bacteria, new bacteria. So this method of reproduction taking place in the filamentous bacteria like Vitriosella species, Leucothrix species, Simonsella species, Saprospira species like the bacteria. So this is all about a vegetative type of reproduction. The vegetative reproduction taking place, as I told you earlier, by the binary fission, budding, and fragmentation. Asexual reproduction by formation of certain spores. So this type of reproduction takes place by formation of endospore and conidiospore. So endospore is considered as a spore which resist and sustain in adverse environmental condition. In an unfavorable condition, the nucleoli of bacteria get duplicated and protoplasm secrete the thick wall around to it, and this thick walled spore is called endospore. So when the favorable condition occur, this endospore again convert into the bacteria. So endospore formation takes place in the bacteria like Clostridium and Bacillus species. While conidiospores, the bacteria like Streptomyces, these are the actinomycetes looking like a fungi, reproduces by formation of conidia. So conidia are formed on conidiospores. So this conidiophore is the reproductive structure. You can look here in the figure. These are present at the tip of a conidiophore. So it produces the conidiospores continuously. A conidia is a single-celled round spherical, and this conidia produces new bacteria. The last type of reproduction is a sexual reproduction. Generally, the bacteria does not record the sexual reproduction. The bacterial recombination is brought together by transfer of genetic material from one bacteria to the other bacteria. The bacteria join together and a portion of DNA from donor to the receiver get transferred, and that is considered as a sexual reproduction. It is taking place by conjugation, transformation, and transduction like methods. So look here the conjugation. The conjugation, in this process, the two bacteria come lie in front of each other. They form a tube. That tube is called as conjugation tube. One bacteria transfer its genetic material into the other bacteria. The bacteria which transfer the genetic material are called as donor, and these are called F plus cell, while the bacteria who receive the genetic material is called as receiver, which is a F minus cell. So in this way, the genetic material get transferred from one bacteria to the other bacteria. Here, the tube formation takes place, that is with the help of pili, and now donor cell is converted into receiver cell, means F plus cell convert into the F minus cell. So this trans conjugation method, transfer of genetic material, firstly reported by the scientists Liederberg and Totem in E. coli bacteria. Transfer of a genetic material. This is very important for the examination. Next, transformation. This method of sexual reproduction was firstly observed by the scientist Griffith in 1928 in the pneumococcus bacteria. The DNA get liberated from the donor bacteria and it is absorbed by the recipient bacteria, and therefore the changes in the structural form takes place. That's why it is called as a transformation takes place. And in a Griffith experiment, the R strain converted into S strain that you have studied in the 12th standard Griffith experiment. Next method of sexual reproduction is transduction. In this method, transfer of genetic material takes place through viruses. So certain viruses like bacteriophages transfer the genetic material from one bacteria to the other bacteria, and transduction takes place. So this is all about the reproduction in the bacteria.